Hello. In this video, I'm going to be talking about drugs and their mechanisms of action on neural transmission. So I'm going to briefly review the structures of the neuron and the synapse, and then I'm going to go over some of the major mechanisms of drug action and how they affect neural transmission at each stage of the process. The synthesis of neurotransmitters, the transport of neurotransmitter molecules, the storage of neurotransmitters within vesicles, the release of neurotransmitters into the synapse, the degradation of neurotransmitters or the breakdown of neurotransmitters, the reuptake of neurotransmitters or the recycling of neurotransmitters that have no longer use. And then we're going to talk about receptor activation. So basically the molecules that the neurotransmitter binds to, and receptor blocking or making the receptor unable to become activated. So all of these are mechanisms of drug action in that they are ways drugs affect the body. So before we go any further, I wanted to make two real quick meta notes about the structure of these videos. So first one, if you have not yet watched that animation of the synapse, stop here and go ahead and do that. Hopefully the way that the announcement is structured makes it fairly obvious that you should watch that video first, but you should watch that video first and then return to this video if you haven't already, because that's going to contain some fundamental information that we're going to build on for the rest of this unit. The primary reason that I am directing you to that video is I think it does a really good job of showing the entire process of the synaptic transmission in a way that really kind of just takes you through each of the steps. And my technological savvy is certainly not enough to create that kind of beautiful computer generated animation. So watch that video first, then go ahead and resume this video if you haven't already. Second quick meta note, this unit and next week's unit are consistently the least popular units in any psychopharmacology class. Most of you are probably not going to find this material quite as engaging as some of the stuff we did in earlier weeks or some of the stuff we're going to do in coming weeks. However, this information is foundational for understanding the mechanisms of drug action. We can't discuss how cocaine affects the human body unless you understand the processes that are being interrupted or altered by the consumption of cocaine and by the ways that the cocaine enters the body. So, I am aware that this material is a little bit technical, that we are getting into some molecular biology and some anatomy and physiology, and that most of you do not find this to be the most interesting unit we cover in this class. Bear with me, do your best to get the terms straight, ask questions as needed, and I promise I am not just covering this unit to be mean. I am doing this because it's a really important set of fundamental processes and structures within the body that we then need to understand in order to understand how drugs affect them. All right, bearing that in mind, a quick review of the structure of the neuron from that synaptic animation you saw. So you can think of the neuron as being shaped sort of like a tree, where it has these branches known as dendrites that, much like the branches and leaves of a tree, take in energy and take in information. So for a tree, takes in sunlight and transforms it and stores that in the trunk. In the case of a cell, the dendrites take in neurotransmitters and action potentials or electrical signals, and they store that information in the cell body known as the soma. The soma is sort of like the trunk of the tree. It is where the nutrients are stored and where neurotransmitters are created. The axon is sort of like the root of the tree. The axon is the long extension from the cell body that travels away from the cell body and carries neurotransmitters and action potentials, or those electrical signals, away from the soma or the cell body. The action potentials and neurotransmitters travel down the length of the axon until they reach the axon terminal. That's the very end of the axon where information in the form of neurotransmitters and in the form of electrical signals is sent out across the synapse. So the synapse is the place where two cells are very close to each other, do not quite meet, and chemical messengers travel across that gap. So here is a close-up of a synapse from the video, and you'll notice that the axon terminal, or the very end of one cell, is very close to the dendrite, or the very beginning of a branch of another cell, but that there is a very, very minuscule gap between the two. Chemical messengers, neurotransmitters, get packaged up within the axon of the presynaptic cell, or the cell that's sending out the message. And then basically the vesicles fuse with the lining of the cell, like you saw in the video, release the neurotransmitter. The neurotransmitter then travels across the synapse where it binds with receptors or specialized molecules on the dendrite of the postsynaptic cell. The postsynaptic cell is the receiving cell. Any leftover chemicals that don't bind to receptors are then taken back up into the presynaptic cell where they get recycled within that axon. Within the postsynaptic cell, those chemical messengers change the electrical signals within that dendrite and make an action potential or an electrical signal to another cell more or less likely going forward. If you didn't quite get all that, or you're feeling a little lost, I encourage you to go back and watch that YouTube video again. I think it does a very good job of explaining all of this. So, how do drugs affect that entire process? Well, there's a whole bunch of different ways, specifically 8 of them that we're going to go over in this class. And as I said, we're going to be returning to this information several times going forward as we talk about specific drugs and specific mechanisms of drug action. So a neurotransmitter or a drug can affect neurotransmitter synthesis. So that's when the neurotransmitter is created within the body of the cell using various basically molecules that then get put together to form the neurotransmitter molecules. So for instance, L-DOPA is a drug that contains one of the molecular building blocks of dopamine, or L-DOPA can pass through the cell membrane, be taken up and used to create more dopamine. And if the individual takes L-DOPA, then one's neurons take in that L-DOPA and use it to manufacture more molecules of dopamine. And if you have Parkinson's syndrome, then you basically experience a lessening of dopamine receptors and a lessening of dopamine effectiveness within your nervous system, which leads to movement disorders and cognitive decline. However, if you take L-DOPA, you experience relief for a lot of your symptoms and can have better quality of life for a very long time because the L-DOPA artificially injects your cells with more dopamine, or basically more of the chemicals needed to synthesize or create dopamine. And it gives your system access to more dopamine for a time. And again, it can give you better quality of life if you are living with Parkinson's. Drugs can get in the way of neurotransmitter transport. So transport is when the neurotransmitter has been created, the cell has made dopamine from L-DOPA, it has made serotonin from tryptophan, etc. And it then transports the molecules of that neurotransmitter down the axon of the cell and gets them ready to release to the dendrites of the postsynaptic cell. Drugs that affect neurotransmitter transport either make that process happen more quickly or more slowly. So, nicotine changes the transport of monoamines, which are neurotransmitters that include dopamine, serotonin, and norepinephrine. And so, these neurotransmitters are basically have an easier time traveling quickly down the axon of the cell under the influence of nicotine. The greater availability of these neurotransmitters in the brain then leads to the increase in energy and the boost in positive mood that is associated with smoking cigarettes. Drugs can affect neurotransmitter storage. So that is when the drug usually sabotages the process of the neurotransmitter molecules getting wrapped up into those little packages of cell membrane known as vesicles, and then attached to the membrane of the presynaptic cell for release into the synapse so that they can then communicate with the dendrite of the postsynaptic cell. So, drugs that affect neurotransmitter storage are ones that cause the vesicles not to form correctly or to be less effective. And so the neurotransmitter, in essence, leaks out of that package and leaks back into the presynaptic cell. It never reaches the synapse and it never gets to communicate with the postsynaptic cell. So, Respirine is a drug that was originally prescribed for heart disease, and it was associated with increases in depression, negative mood, and other mental disorders when individuals took it long term. What was eventually discovered is that Respirine causes the vesicles that transport especially serotonin to leak. These little packages don't form correctly, and so the serotonin basically gets out of the vesicle, remains in the presynaptic cell, never gets transported across the synapse, and never reaches the receptors of the postsynaptic cell. So serotonin is being made, it's being transported, but it can't get out of the presynaptic cell very efficiently. So there's just less of it to go around. And that was why it was leading to these mood disturbances. Drugs can interfere with neurotransmitter release. So that's where the vesicle carries the little package of neurotransmitter down to the membrane of the presynaptic cell. The vesicle attaches to the membrane, opens up, lets out its package of neurotransmitter into the synapse. The neurotransmitter drifts across the synapse and attaches to the receptors of the postsynaptic cell. And if the drug interferes with neurotransmitter release, then it can cause the process to be sabotaged in that too much of the neurotransmitter will be released. So the presynaptic cell has those reuptake receptors where it is basically taking back some of the receptor it releases as a way of monitoring how much of the neurotransmitter is floating around the synapse. If too much of the neurotransmitter is floating around the synapse, then the presynaptic cell stops releasing any more neurotransmitter and the signal stops going out. However, if you have a drug in your system that interferes with neurotransmitter release, oftentimes those signals are not going through, that feedback loop is not working properly, and an excess amount or too much of the neurotransmitter is released into the synapse, which can then lead to very serious problems with neurotransmission. These tend to be very harmful drugs. So, botulinum toxin causes an increase in the release of acetylcholine. Acetylcholine is a neurotransmitter associated with movement. It's actually found in the muscles of the body as well as in the brain, which is part of the reason it was the first neurotransmitter ever discovered. And having too much acetylcholine in the system leads to paralysis. So botulinum toxin actually has led to many fatalities. It was especially associated with fatalities during the Spanish-American War because it would grow in improperly canned foods where early canning processes did not make airtight cans. So the toxin would grow in the food. Soldiers in the Spanish-American War would eat the food, and then they would develop a deadly case of paralysis. This toxin is believed to have killed more American soldiers in the Spanish-American War than the Spanish did. So this was a major source of death among, again, individuals whom they were having an excess of acetylcholine flood their brains and their muscles because the toxin led to a sabotaging of that process of the correct amount of release or the correct rate of release of acetylcholine. Nowadays, it's actually used for Botox treatments where very, very small amounts are deliberately injected into the muscles of the forehead. This paralyzes the muscles of the forehead and reduces the appearance of skin wrinkles on the surface of the skin, especially as they develop naturally on the forehead and around the eyes and the mouth with the natural process of aging. Neurotransmitter degradation is basically the cleanup process where the presynaptic cell releases enzymes, and there are enzymes sort of floating around in the fluid of the synapse. I'm oversimplifying here, but hopefully you get the idea. And those enzymes basically find stray neurotransmitter molecules floating around and break them down and reuse the materials that went into the neurotransmitters. If neurotransmitter degradation does not function correctly, then those neurotransmitter molecules are floating around for a longer period of time, and they're more likely to meet the receptors of the postsynaptic cell and more likely to activate those receptors. So the neurotransmitter is more effective when neurotransmitter degradation is being interrupted. So cocaine has actually many different effects on many different neurotransmitter systems, but one of the known effects of cocaine is that it prevents the breakdown of those monoamines, dopamine, serotonin, norepinephrine, and in the process, it causes dopamine, serotonin, and norepinephrine to be more effective in that basically they're not breaking down, so they have more time to float around and end up in the receptors of various postsynaptic cells. So they are more effective, and that leads to the boost in energy and boost in mood and sometimes manic state associated with use of cocaine. And then neurotransmitter reuptake is when, as I alluded to, the presynaptic cell basically recycles the molecules of the neurotransmitter. So it's released the molecules of the neurotransmitter. Some of them have ended up on receptors of the postsynaptic cell. Some of them are still just floating around in the synapse. They might get broken down by an enzyme, or they might get taken back up into the presynaptic cell, where they get recycled back into that axon, and the next time an electrical signal goes out or an action potential goes out, those molecules might get packaged up and then reused to send out another signal across the synapse. Drugs that change the rate of reuptake make the neurotransmitters more effective because they have more time to have their effects and have more ability to reach the receptors of the postsynaptic cell. So you may have heard of SSRIs, which are selective serotonin reuptake inhibitors. So they are exactly what the name sounds like. They prevent serotonin that is floating around in a synapse from re-entering the presynaptic cell, and in the process, they make serotonin more effective at regulating mood, sleep, and diet. So Prozac is the brand name for fluoxetine, which is an SSRI that is widely used to treat a variety of mental disorders, including depression and certain anxiety disorders. The way that it does that is through basically causing serotonin as it's present in the brain to be more effective than it would otherwise be, and in the process leads to better regulation of mood, sleep, and appetite. So drugs can affect the process of receptor activation. So now we've moved to the postsynaptic cell. We're talking about that dendrite that's receiving the chemical message. or sometimes it is not receiving the chemical message because a drug got in the way. So when a drug is affecting receptor activation, it's when a drug has generally a similar chemical structure to an actual neurotransmitter, and then it mimics the neurotransmitter by attaching to that receptor instead of the neurotransmitter itself. So, for instance, opiates, we now know actually work through mimicking a naturally occurring neurotransmitter known as endorphin. And what happens is that under normal circumstances, the presynaptic cell would package up an endorphin, generally under conditions of severe distress or pain, as a numbing agent. It would send it down the axon, it would be released. But if the individual instead takes, say, morphine or Oxycontin, then what happens is the tiny molecules of morphine travel to the receptors on the various postsynaptic cells, and they actually slot into that receptor. And the postsynaptic cell perceives that it has received an endorphin signal, and in the process, it deadens pain. And so endorphins occur naturally within the body to deaden pain, and then morphine can mimic the action of endorphins through activating the receptors for endorphins, and again, basically slotting into the place where the endorphins would normally go on the postsynaptic cell, and then activating the postsynaptic cell's receptors as a result. Drugs can also block off those receptors entirely. So the metaphor often used here is if you imagine you have a lock, and instead of putting a key into the lock, you jam a nail file in there and you break off the nail file. Now, nobody can use the lock, nothing can get through the door. So drugs that basically block a receptor mean that nothing can get in through that receptor. The receptor can't receive signals, and so the presynaptic cell can release all the neurotransmitter at once, but that signal's not going to go through to the postsynaptic cell because the postsynaptic cell can't receive those molecules. So, for instance, naloxone also works on those endorphin receptors. But the point of naloxone is that it, again, jams those receptors shut so that they can't receive any further signals. And so it protects people from overdoses of opiates because it jams shut those endorphin receptors, so the individual can then basically stop receiving any kind of signal from Vicodin or the heroin that they may have overdosed on, and in the process can then survive the overdose because the heroin stops having its effects on the brain with the naloxone in the system, again, basically blocking the receptors that the heroin would otherwise be using. You also probably have realized how drugs have their long-term effects based on all of these short-term effects that I've been talking about. If you are consuming a drug like Respirine again and again and again that makes your serotonin vesicles unable to function, then over time, the cell is going to start producing more and more serotonin to try and get the message through. And it's going to have more and more basically inefficient processes to try and get around the repeated effects of this drug. If we talk about something like heroin, so generally what happens is the individual uses heroin, the receptors for endorphins get activated, and the body pretty quickly realizes that the receptors are being too heavily activated. So it will sometimes create more receptors to cope with that. It will sometimes decrease the sensitivity of receptors to cope with that. And in the process, the individual becomes more and more tolerant of heroin, needs to take more and more heroin in order to achieve that endorphin effect, and unfortunately runs an ever greater risk of overdosing on heroin.